Okay, so the first instruction in our lab protocol is you're going to be starting with 50 grams of clean spinach leaves. The protocol says make sure that you refrigerate them for 15 minutes, but they're already going to be in the refrigerator, so we're not going to worry about that part. So here we have spinach leaves, and remember from the lecture that the stems don't have as much of the chlorophyll, don't have as much of the chloroplasts and the thylakoids, so they don't have as much chlorophyll as the rest of the leaf. So remember, you're going to be removing that part of the stem so that you're getting just the greenish part of the leaf. And as you're doing that, you're going to make sure that you are washing all the leaves. Now, you don't have to wash a single leaf at a time like I've done, but make sure that you get all of them washed, because spinach is a filthy plant. And then as you get a bunch of them, make sure that you're just sort of patting them dry so you don't have too much extra water on them. After you've done that, you're going to grab a bunch of leaves and you're going to weigh them on the balance. So over here, we have our balance, and you want to make sure that you put a weigh boat on it and that you tare the weigh boat. So to tare, you push down on this particular balance and wait till it says 0. Then after it says 0, you add leaves until you get up to a 50-gram mass. So in this case, this is a little bit more than 50 grams of washed and mostly dried off leaves, 49.62, it's pretty close to 50. One of my favorite sayings is this is biochem, not analytical. So in this case, you don't have to get to 50.00 grams. You just have to write down how many grams you had, because that is lab data. Then you're going to come over here with the leaves and you're going to put them into the blender. So you can see I already have some leaves in the blender, and if you read the protocol tells you to add 120 mils of STN to the blender. Reserve a little bit back, just a little bit, for rinsing the blender. I'm reserving maybe 10 mils back. You're going to put the lid on the blender, and then you need to blend it for a good 15 to 30 seconds until everything is blended in the solution. So 15 to 30 seconds is not how much time you have it on. 15 to 30 seconds is when the leaves are actually going around in there. So you'll start by blending, it'll be loud, and then stop, and you're going to take the lid off and push some of the leaves down. Don't ever do this with your fingers, and don't ever do this when the blender is going. And I say this, you will hear many warnings from me in class, but I give warnings because I have seen stupid behavior from graduating seniors. So that's where a lot of these warnings comes from. So trying to push all the leaves down, and when you have all the leaves pushed down, trying again to blend. When all the leaves are now in solution, then you can start your 15-second time. So now you have, and it's important to put a lid on because otherwise you're going to be getting plant pieces everywhere. I even see them on the camera that was being used to film here. So it's going to look like a smoothie. So basically you've just made a spinach smoothie. So the next instruction says that you're going to filter it through muslin, and this is MiraCloth, treated with EDTA. And this is actually in a funnel, and the funnel is going into what we call a GSA bottle. I know your instructions say put it into SS34 tubes, but on the assumption that you have multiple groups doing this at the same time, we don't have enough centrifuge capability for everyone to have individual multiple SS34 tubes like you would if you were doing this in a lab just by yourself. So we've modified the procedure a little bit to say, okay, every group's just using a GSA bottle. So you're going to pour this in and you're going to let it filter through. Give it some time. It'll keep filtering. Pour more in as you're able. And what it's filtering out, all those cells that are now broken from the action of the blender, the cells broke and released the chloroplasts into the solution. Chloroplasts, remember, are an organelle. So all of those chloroplasts have been released into solution. There's still fibrous material in there though. So the whole cells, most of the whole cells, some might get through, most of the whole cells and the fibrous material are being held back by our mirror cloth. And what's coming through is we have some dirt particles. Remember from the lecture, we have a few whole cells, and we have a lot of chloroplasts, and we have some other organelles as well. So remember, when we are finished with this procedure, we're going to be doing 3 differential centrifugations. And so if you don't remember those 3 differential centrifugations, make sure you refer to the lecture as to why we're doing each of those 3 differential centrifugations, what we are separating at each point. So you can see now I have some space. I'm going to be pouring a little more in, letting it keep filtering through. And now I have my extra 10 mils or so of solution that I have reserved, putting that in, just like you did in organic chemistry, using it to maximize your yield. Trying to get as much as we can out. Not necessarily 100%. So it's filtering through, and as it's filtering through, you can pick this up carefully, and you can apply some gentle pressure to the outside to get some extra solution out. Don't squeeze too much. If you squeeze too much, you will break the mirror cloth, and all those contents you've been trying to separate will go plunking down into your bottle. So just some gentle pressure on the bag, trying to maximize your yield. So now you have your solution containing chloroplasts and a few other things that we're going to be separating from it. You're going to be balancing this in a centrifuge across from another group so that you have the same mass on both sides of the centrifuge. If for any reason there are an odd number of tubes, then one person, one group needs to get a similar bottle, weigh this one with the lid on it, make sure with the lid on it, and fill it up with water or with STN solution so that it is the same mass as your other tube, and then you can spin them across from each other in the centrifuge. Okay, so in our protein purification lectures, we talked about different types of centrifuge rotors. And these are some examples of fixed angle rotors that we have, that they're going to spin our samples at a particular fixed angle. This is an SS34 rotor. And this one fits those GSA bottles that you just saw us putting our spinach sample into. It's actually called, because they change the names sometimes, F14 6x250Y. So what you see on the lids here is you see some controls, and these controls have 2 parts to them. The bottom one, when you're putting it on the centrifuge, has an on in this direction and an off in this direction. Now please note, this is not lefty-loosey, righty-tighty. It's the exact opposite of lefty-loosey, righty-tighty. So if you're ever not sure, please just look at the instructions on the top, which tell you which way you need to turn it to either put the lid on or get it off. So once you have your samples in here, and we have our water balanced because we're doing this as a single without another group, you have them opposite from each other in the rotor. So these are opposite each other and they weigh the same amount, I've put them on the balance to make sure they're within about 1 gram of each other. So then we're going to put the lid on, and the first lid that goes on, the bottom one, now remember this is on, so I want to be putting my lid on, and that's going to be putting this on. And you can, when it's in the centrifuge, if it's spinning, you can feel free to hold this part. Don't hold this part though and try to spin this. You're going to hold this and spin the lid around. Trying to hold this and spin this around, it's a little bit of extra tension that you don't need when doing this. So finger tight, not cranking the heck out of it, just finger tight so that you can go, oop, I can't get it any tighter. Don't go, and I've had some students do this. Okay, I've got it on as tight as I can. Well, that's nice, but you're never going to be able to get it off now. This bottom one fixes the lid to the rotor. This one fixes the actual rotor to the inside of the centrifuge. So please look right now at the inside of the centrifuge and see how there's this triangular center portion. And this triangular center portion is going to fit right inside here. So we're going to put this on here, and you can have the rotor already set in there. We only had it out for purposes of the video. And you must, absolutely must, fix the rotor to the centrifuge itself. So again, just until it's finger tight. So then you can close the lid. Now, we are going to make sure that it is set for the correct rotor, and we're setting the correct speed and the correct time. So every centrifuge is a little bit different in terms of its controls, but this one, as soon as you close the lid, there's a little select rotor that's blinking at you right there. So you can read select rotor right there. So to select the rotor, you push the rotor button right down here. And that rotor button then puts all the different rotors which people use in up on the screen here. So remember, it's F14 6x250Y. That is our particular, just F14 6x250Y. That is our particular centrifuge rotor. So we want to make sure that we're selecting the right one. And if you don't see your initial rotor, you can move the screen down and around until you do see your correct rotor appear on there. Okay, so here we have our F14 6x250Y, and this is number 48. So you would put a 48 in here, 4, 8, and then enter. And so the number appears here. So 4, 8, enter, and then you get to your screen where you can actually set the speed and the time. So you can do this by using the cursors. So to set the speed, you can move the cursor till the speed button is selected. For our first spin, we're going to be spinning it at 3,300 RPM, and it adds a 0 for you. So pushing 330 and then enter. And then if you need to, you can move it. You can use the cursor button to move it to the time. And we're going to do this for 7 minutes. And then it's very important to push the start button. If you don't push the start button, it will never, ever spin. So once you push the start button, it will start to accelerate. It will tell you how fast it's going right up here. And this particular model will play you a nice little song when it's all finished. But if you're in the other room, you're not going to hear it. So we'll pick up this video when the centrifuge is down. Okay, so when the spin has completed, remember that you need to unscrew both of these. You're going to have to unscrew the top one in the off direction before you can remove it from the centrifuge. Although you don't have to remove it from the centrifuge, but it does need to be loosened from the centrifuge before you can do the second one, which gets the lid off. So when you finish screwing this one off, then you'll be able to remove the lid. After you remove the lid, you'll be able to take your sample out. And I don't know whether you can see, but there's a pellet just along here on the side of the bottle. But you want gravity to work in your favor. So when you are carrying it, it needs to be pellet side down. Because if you carry it with the pellet up, you're going to be sloshing the solution into the pellet. You're going to be prematurely loosening your pellet, and you don't want to do that. So now we're going to carry it into the next room, and when the video picks up again, I will remove all of the solution off of the pellet. And so when the video picks up, you will see just the pellet. Okay, so the pellet is very loose, so the pellet can be a little bit watery, so don't get panicked. When you see dark green solution, if you're pouring it off or pipetting it off, don't pipette off any of the really dark green stuff, even if it's sort of loose, because that is part of your pellet. So when you have the loose part of the pellet, and maybe with some still solution in there, you're going to put 35 mils of your STN solution back in there. Now remember, the very first spin on here, the very first differential centrifugation, you were spinning, and what's in the pellet is the chloroplasts and everything larger. And in our second differential centrifugation, we want to get rid of the everything larger pieces. So we are pipetting the solution up and down to resuspend the solid parts of the pellet. So if you have solution in your pipette and you have a pellet, you can pipette it onto the solid part, and that will help it all dissolve. After your pellet is completely resuspended, you're going to be spinning it again in the same centrifuge rotor that you did before. This time you're only going to be spinning it at 1600 RPM though. It's going to be the exact same process for putting on the lid, setting the timer. You're only doing it for 1 minute because the purpose of the second spin is to just separate the chloroplasts, which are now in solution, from anything that's larger than a chloroplast. So you want to just have a very short spin time so that all those dirt particles and whole cells come down and pellet out, and that you can just pour your supernatant away from those. So that very short 1 minute spin at 1600 RPM is just to get rid of anything larger than a chloroplast. After the second spin, you will have a very small pellet that will consist of whole cells. Remember, anything larger than a chloroplast, and maybe some dirt particles. So for your third spin, you just want to pour it into an identical bottle, leaving the pellet behind. So here we have our pellet still up here, and we have our solution now with our chloroplasts in it. So remember, the first spin, we got rid of anything smaller than a chloroplast. In the second spin, we got rid of anything larger than a chloroplast. So what we have in here now is just chloroplasts in solution. So we're going to do a third spin that's identical to our first spin, 3,300 RPM for 7 minutes. And during that time, we're going to be pelleting down all of our chloroplasts. And at the end of that spin, we will have a pellet that consists of our chloroplasts. Okay, so now we're finished with our third spin, and you'll notice that the solution is a lot lighter here than it was before. And that's because, remember the first time we were getting rid of anything that was smaller than a chloroplast, then we got the second spin, got rid of anything larger than a chloroplast, and now we've just pelleted our chloroplasts. So now you can see our pellet right here with all of our chloroplasts and a much lighter solution here. So I'm going to pour off the pellet so that we only have a pellet remain. Pouring very carefully. So now you can see our pellet. It's still very loose and going to come out. And now we're bringing it up in 2 mils of STN. So here we have our STN solution, and it's been sitting on ice, so it's still cold. We're going to add 2 mils of STN to the pellet. And we're going to follow the same procedure to resuspend it in 2 mils of STN, just like we were before, pipetting up, placing the tip near the pellet, and pushing down to get it all resuspended as quickly as possible, using a little bit of force to resuspend that pellet. You cannot vortex when you have whole cells. Vortexing will destroy the membranes of the whole cells. These are not stable enough for you to for you to vortex. And remember, these aren't even whole cells. These are just organelles that are inside the whole cell. So if you were vortexing at this point, you would be completely blowing apart all of your chloroplasts, and you don't want to do that yet. So now you can see our solution's becoming much darker as we resuspend everything in that 2 mils of STM. We're almost there. Okay, so we have resuspended our sample in the 2 mils of the STN. I know the protocol says 2 to 4 mils, but it's always better to use less. So we've resuspended our sample in 2 mils of STN. And before you add the water to it, you want to make sure you take a sample. So you need to take a 0.2 mil, a 200 microliter sample of it. So you're going to come down here, take 200 microliters out of it, and you're going to add it to a 15 mil tube. Now notice this is a change in the protocol that we're doing from the lab manual. And we're doing this because the spectrophotometers do not have 5 mil spectrophotometer tubes anymore. We're going to be using our plastic 1 centimeter 1 mil cuvettes. So this is a change in protocol. So now we're going to say, what's the total volume? Because our next step in the procedure is to take our total volume of solution and multiply it by 10, and that's how much water, how much DI water we're going to have to add so that we can do an osmotic shock procedure. And again, refer back to the lecture. So our total volume here is 3.8 mils. And you were using a 10 mil pipette, and it reads both directions, so 3.8 mils. So we know that we want 38 mils of ice cold water. So we're going to pour out a little bit of this that I had till we get to 38 mils of ice cold water. Almost there, a couple more. Okay, that's 38 mils of ice cold water. So now we're putting our chloroplasts in the ice cold water. And now we're going to put them in SS34 tubes. And again, you want to be able to balance the SS34 tubes. You don't want to try to fit them all in one. You want to divide them between 2 equal tubes so that they're the same height. Okay, so now we have 2 equal volumes of solution in our SS34 tubes. We're going to be using an SS34 centrifuge to spin. Same procedure as before for spinning and for choosing which rotor that we're going to use. So you've seen all these before. Same procedure for setting the speed. Everything's going to be the same, but we're going to be using an SS34 rotor instead of the F14 6x250Y rotor. We're going to be putting these in opposite, and we're going to be spinning them for 20 minutes at 12,000 RPM. Always remember to have your protocol to refer to so that you don't make mistakes.